Well, welcome to another episode of In the Raw at Pro Raw with me, Andrew Locke, and today our special guest, special researcher, PhD candidate, Bobby Shamim.
Thanks for
Bobby,
having me. Yeah,
thanks for coming on
pleasure
in.
to be here.
Well, you take on a very different world to most people.
Yeah.
You take on a world that's a bit like the Fantastic Voyage. Every day you go to work and you get in your little special thing and you end up down a microscope.
Yeah, that's right. Just looking at kind of the mechanisms that underlie why you get big and strong, why the training might be more effective in certain circumstances than others, and how nutrition can promote the adaptations that you might be making. So yeah, trying to understand what are the contributing factors that underlie.
And what took you on this journey? How did it all start? What stimulated you? How did you get into undergraduate? What happened?
Yeah, I mean, it's really good to reflect on that actually, because when I was an undergraduate, I didn't really know what I wanted to do. I've always been interested in physiology, human behavior, just biology in general. And I studied at UC Davis in California, and they provided a really good program called exercise science, exercise biology back then, where really I just kind of exposed myself to a lot of different elements of human physiology. I didn't really know what I wanted to do, but I was just open to trying new things. I picked up a bunch of internships just to kind of learn. I mean, that's the best way to do it, is if you're not sure what to do, just do everything, filter it out, and go from there. And one of the things that I got quite intrigued in after taking a course from one of my professors was trying to look at the research behind human performance. Yep. And kind of got introduced to more of the biochemistry side of things, and found myself in the lab, just learning basic techniques and trying to measure certain proteins that might be present in the body, certain gene expressions in muscle, things like that. And I kind of thought, the research is really quite stimulating. It's exploratory. I like knowing how things work. And so that kind of, lit a good flame for me to pursue the academic side of things. And then I carried on and I went to the University of Birmingham in the UK, where I studied a Master's of Research. And that was trying to apply some of the biochemistry into training interventions. So looking at, leg strength after a 10-week-long intervention, and just seeing how we can adjust training variables to improve strength and muscle hypertrophy. And from there, I completed that and came over to Australia to start my PhD in exercise sciences at the Australian Catholic University. And here, at the moment, I'm studying changes in gene expression and how that affects our adaptations to exercise, and how nutrition, specifically protein, can promote those adaptations.
Very interesting.
So, yeah.
And with your own weight training journey, when did you start lifting? Because you're training down at South Melbourne, PTC?
Yeah, so trained down at PTC South Melbourne with Alex Deacon. Great facility, great group of athletes there as well.
Super people, yeah, great coach.
It's awesome. As far as my own training, again, it kind of started off like any gym rat would. Like I was always intrigued by human physique, building muscle. So just kind of got into the gym at a young age. I mean, I started when I was 13, 14, just kind of doing bodybuilding stuff, bicep curls, and a lot of bench press, I can tell you that. But no, it was just trying to understand myself, proprioception, trying to see how big and strong I could get. And then actually one of the internships that I did pick up at UC Davis was with the strength and conditioning program for the Aggie football team.
Yep.
And working there, I kind of got more intrigued about strength and building strength, and tried to apply that to myself, and slowly got more into the powerlifting side of things with no real structure. It was mainly just competition with one of my colleagues who could lift more. Unfortunately, that does lead to injuries, which is why I've found myself here. But you live and you learn, right? It's all about the learning process. So yeah, that was kind of the intro to me. And then after I finished my master's, I'd moved back to San Francisco, and I became privy to Dan Green's gym, Boss Barbell Club. It was just down the road from me, which I was completely oblivious to. So I started training there, and I learned a lot, gained a lot of strength there as well. And then, came down to start the PhD, and for a while I was just sort of training in our laboratory, really. We have a strength facility there, and I was just training by myself.
It's actually a pretty good facility too.
Yeah, it's not a bad facility. It's got everything we need, but training by yourself can get quite lonely. So yeah, I got turned on to PTC South Melbourne, and yeah, haven't looked back. Great place.
And now with the research for yourself, you're finding some superbly interesting things that follow your passion, which is how do muscle cells grow?
Yeah, that's right. So one of the big questions is how do muscles actually grow?
Yeah.
We know that there are certain stimuli, for example, weightlifting. We know that protein, amino acids, these can all facilitate muscle growth. But as far as the underlying mechanisms go, it's actually quite a big mystery. We know that there are a few key molecular transducers. So the way that you can think about this, and some of the work that I do, is when you drive your car, for example, the car can be acting as like the function. But when you come up to a stoplight and there's cars coming from all different directions, the stoplights are what are saying you can go, you can wait, you can halt, yield, slow down, whatever. So that's essentially the same thing that's going on within your muscle after you provide a stimulus like exercise. There's certain signals that are turned on, there's certain signals that are turned off, and these signals are multiple and numerous at once, simultaneously, or they can be antagonistic, they can run in parallel. So it's quite chaotic actually at the molecular level, which we don't really think about because we're not molecules. Although it is important to understand what's actually going on so that we can try and optimize and that we can try and prescribe appropriate means of training intervention or rehab in your scenario to try and recover the muscle to the highest quality.
So it's very interesting. We obviously mostly you'll read as far as the most basic work will come to us. We're going to talk about concentric exercise, eccentric exercise, isometric exercise. And you've actually been looking at the responses, even under the microscope, to each of these as an individual influence, as a variable.
Yeah, that's right. So one of the really interesting things is we know that with different types of contractions, there can be more or less damage. And trying to understand what are some of the repercussions of the amount of damage at the molecular level, and what are some of the mechanisms that can contribute to the recovery is quite a topical issue at the moment. And my research in particular is quite focused on some of those factors, in particular the muscle stem cell, known as a satellite cell, and how that contributes to the recovery process. So we know that the muscle itself has a nucleus, and the nucleus is what contains your DNA, and the DNA is what essentially encodes for who you are.
Yep.
And when you interact with your environment, we're here at ProRaw, so when you lift big heavy weights, and when you work with Andrew and he gives you good rehab, you can stimulate the expression of those genes in a certain pattern that can facilitate and promote the adaptation that you're making. And then from those genes, eventually they get turned into proteins, which are the functional unit that then carry out and execute whatever the action might be or the recovery. But we know that the nucleus by itself sometimes needs assistance. And we can get that from the satellite cell, and the satellite cell, as any stem cell does, essentially gets incorporated into that tissue. And so you've then doubled the nuclei that you have. So maybe you had one in that particular muscle cell, and now you might have two. We know that the muscle cell is multinucleated. It has 5, 10, 15. It can all depend on the size of it. But essentially what you're trying to do is you're trying to increase the transcriptional machinery that you have so that you can increase the upregulation of gene expression and proteins and try and facilitate and promote the adaptation more.
It's absolutely outstanding. Like, I mean, we would look at our basic understanding. We talk about fast twitch fibers, slow twitch fibers.
Yeah.
But there's more to it than that, isn't there? There's a lot more grey area.
There's a lot more grey area. It's still early days. I mean, we've known about satellite cells. And when I say we, I just mean the scientific population. We've known about them since the 60s. We didn't really know what their function was, but until recently, now we can use newer technologies. The great thing about doing science now, it's really exciting with all the biotech. We can try and apply some of those feats of engineering into biology, and we can try and understand at a more broad range how the satellite cells work, or not even just satellite cells, but just really any process. So whereas we used to just look at single targeted approaches where I might say I have my one favorite molecule, it might be myostatin, for example. We know that one's quite heavily involved in regulating muscle size. So whereas back in the day we used to just look at one single protein or one single gene of interest, now we can use high throughput sequencing technology to be able to screen for tens of thousands of different transcripts or proteins at any given time. So it's a really exciting time to be doing all this type of research.
There's so much more that you're seeing now than would have been 10 years ago.
Yeah, that's right. And the speed that you can do it in, and the computational biology that comes along with it to be able to design predictive models about what might occur to a given stimulus yeah, that's what's driving me forward.
And for your own
getting
training.
excited.
I
And for
mean,
my own training,
what
yeah.
do you look at what your research shows you, and do you find if you're looking at a power lifter approach, which is saying let's put on some lead muscle. When you look at your own training, you look at your sets, your reps, your volumes, your recovery times. Do you think about your own research when you're looking at those sort of variables as well?
Yeah, absolutely. I think the one that I'm most interested in is actually the change in the muscle cross-sectional area relative to your change in strength. So one of the big challenges for powerlifting in particular and other weight-restricted athletes is we need to be as strong as we can in a set weight class. And we know one of the great ways to get strong is to increase our muscle mass, like you said. But occasionally if you increase your muscle mass, to quite a large magnitude, that might tip you over the weight class you need to be in. Or for example, we know if you need to cut down, well, you're potentially going to lose a bit of muscle mass when you do that weight cut. So for me, one of the really interesting things to understand is how do we increase strength without actually changing the muscle size? And so you need to actually look at factors that go beyond just the myofibers, just the actual muscle itself.
Beyond the contractile mechanism.
Beyond the contractile mechanism itself, and you need to understand the supporting structures, the cytoskeleton, the extracellular matrix, things like collagens, things like your tendons that are going to be actually transferring the force to be able to truly understand, well, what are all the contributing elements to strength? So yeah, I mean, we know you lift weights, your muscle gets big and strong. How it's occurring is, it's kind of a roll of the dice.
And we were chatting a bit about how ethics have changed over the years.
Yep.
And you were mentioning an interesting study that involved 4 groups. So perhaps you can enlighten people about the results there.
Yeah, so
Back in the 90s?
Back in the 90s, the first author's name was Bassin, and I can link you to the paper and you can share it with your followers because it's a really cool paper. I mean, that was one of the papers that I read during my undergraduate, and I just thought, man, this is like it's cool. That was like my introduction to muscle physiology and biology itself. But essentially, the premise of the study was to just understand the action of testosterone in regards to muscle mass. And so the premise was that there was 4 groups. There was one group that was just a placebo control, and they didn't do anything. There was another group that had a placebo supplement, and they did some resistance training, so some strength training. There was a third group that had testosterone supplementation with no training. And then there was a fourth group, which was kind of probably the best group to be in, was the testosterone supplemented group with the strength training. And as you might predict, the group with testosterone and strength training saw the best response to the resistance exercise. They saw the greatest increase in lean mass. The group that just did the strength training with the placebo, as you would also anticipate, saw some good increases in lean mass. The really interesting thing was the group that just took testosterone, didn't touch a weight, saw the same increase in lean mass as the group that just did strength training without any testosterone. So, I mean, it kind of raises the interesting questions as, okay, well, what are the mechanisms of action that testosterone is working through? Because if you can just take testosterone by itself without any contractile stimulus, it's kind of indicating to you, okay, well, it's got its own separate pathway potentially that it's working through. And we know that if you do it in conjunction with strength training, it seems to add on to the benefit. So they're potentially working in parallel with each other. But one of the things that is quite difficult to study with exogenous supplements now, steroids if you will, is that ethically it's quite difficult to now give research participants those levels of illicit substances that they might take themselves anyways.
Yeah, it becomes a problem, doesn't it? So really we can look back at those sort of studies, which just give us a start to realise there's multiple pathways to muscle changes.
Yeah, that's right. There's multiple pathways, and a lot of the work that we do and that we try and model is done in model organisms. So things like mice and rats, and the reason there is because we can actually just control the environment a bit better. And in doing that, we can remove some of the variability that is inherent with humans. We're all different. Our genetic makeup is different. Our environments are vastly different, just depending on our day-to-day activities, where we live, our nutrition. And so trying to understand, or at least remove the environmental aspects to understand the underlying physiology is a great challenge. But it does provide us a lot of unique, I guess, future directions that we can apply to people, to humans.
Yes, as you were saying, if people work on simply increasing the volume of the muscle itself, but they aren't addressing tendon, collagen, remodeling and strengthening as well, then there's probably more chance that they'll tear a muscle.
Yeah,
or tear a tendon.
That's right as well.
Producing more force without having something that can take the force.
Yeah, and so, everyone's training, and for good reason. I mean, we all think muscle, 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 muscle. Lift weights, well, what's generating the force? It's the muscle. But as you've raised the point, is that the recovery and the actual application of movement is not just muscle itself, but there's all the supporting structures that we do need to address in our training, in our rehab, in our recovery.
Training to strengthen a tendon is different to training to increase lean muscle simply by itself.
Yeah, that's absolutely right. You're training two different elements it's the same beast, but it's just different sides, and you need to address all of them within your training. And so we know that, for example, if you want to increase muscle size, stretch along the muscle is really nice, and we can do it through concentric loads, we can do it through eccentrics, through isometrics, but the main thing that we understand is, as far as muscle growth goes, it's about fatiguing the muscle. So it can be with a lighter weight, it can be ballistic, it can be slow, it can be eccentric, concentric, whatever. As long as you're getting to volitional failure, that tends to be the main stimulus to grow.
Volitional failure.
Yeah.
Yep.
As far as the more inert tissues, the connective tissues, there's quite a lot of research looking at the tempos. It seems to be that the more ballistic movements are going to, make the tendons more brittle. And brittle tends to lead to injury, which is not ideal. So what the research kind of implies is that the tendons respond better to slow eccentric movements, so it can handle heavy loads, but it just prefers it at a slower tempo. And so it's really important to understand the different speed of movements, and not just the load. I mean, load's obviously very important for programming and peaking and things like that. But it's also quite important as a tool itself for rehab processes. So,
Yes, tendinopathies now obviously has become a hotter topic because we're understanding more about them.
Yeah, that's exactly right. And, as far as my field, we're interested in understanding the nutrition that can maybe assist in, or act as a sort of remedy, if you will, and help prevent some of the tendinopathy.
Like we were talking about the famous bro science anabolic window.
Yeah. So the anabolic window is always a good one, because I'm sure, as everyone has heard, the anabolic window is immediately after you put the last weight down of your last set. But actually, what the research shows is, if you're a novice, maybe you're brand new to lifting, and you're very concerned about the anabolic window, the great news is your anabolic window is actually up to 48 hours in the untrained state. So the body is quite receptive and sensitive to nutrients and growth factors after the initial bout of exercise.
About 48 hours.
Yeah, and as you get more trained, as your body becomes more refined and more resilient, so we're better able to deal with perturbations, to stimuli coming from weight training or whatever it might be, the window gets a little bit shorter, but it still gives you 24 hours. So the anabolic window is probably up to 24 hours in well-trained individuals, and the reason that is because the muscle itself is more sensitive to nutrients and recovery and growth factors, and you have other elements and components that when you train, so your muscle doesn't just get bigger and stronger, but it also increases the blood flow. You get angiogenesis, you get more the capillary beds that deliver nutrients, you have better perfusion. We know that you have cardiovascular benefits that come along with not only weight training, but obviously you're running, you're cycling, if you do any of that for your recovery. As a powerlifter, we probably neglect it a bit, but for the general population, it's extremely important that you do both modes. So we know that as you increase these adaptations and these different avenues where you can support the muscle, then your muscle is actually just more sensitive all around. So yeah, you kind of...
You don't need to have that protein drink in that first
No, you
30
don't.
minutes
No,
after.
you really don't. We know that, yeah, in the initial phase, right after, you have the greatest blood flow to the muscle at the time. You're the most sensitive to nutrients for recovery. But realistically, you know, if you get a meal in the next few hours after your training, you'll be fine. It can even be strategies, probably not great if you're trying to increase muscle size, but there are strategies where you might do train low for certain circumstances if you're trying to increase, you know, components of endurance. So potentially if you're maybe more on the, I guess, team sport elements, or potentially CrossFit now is super popular, you know, and that requires a lot of endurance capacity. So we know that there are strategies where you might actually want to deplete some nutrients and be training in a bit of a fatigue state, because it actually just adds to the stress of the training. And then when the time comes, you would then increase the nutrition appropriately. You'd have sufficient carbohydrates, fats, and proteins to assist and promote the actual performance. So it is a really fine balance between, do you always have the gas pedal fully to the, you know, pedal to the metal at all times, or do you need to be strategic and say, well, actually, there's times where I need to make this more challenging for myself, whether it be through depleting nutrition or through a different stimulus, like an eccentric, like heavy eccentrics. They're all different tools that you might have in your tool belt.
How well can the body tolerate heavy eccentrics as a recovery mechanism? How frequently could people do them?
Yeah, so the frequency is a major issue. As far as the eccentrics go and the recovery goes, the research is not conclusive at the moment, but we know that eccentrics that are quite damaging.
You're saying the most damaging to the
most
muscle cell.
Yeah, as far as contractions, that's the most damaging that you can place upon it, and the damage can last for up to 96 hours after. So it kind of raises the question, well, eccentrics are a great stimulus for muscle growth, but also it's a great stimulus because it causes a lot of damage. So if you're constantly using eccentrics as a tool to grow, but not allowing the time to actually recover and facilitate complete growth, then you might be shooting yourself in the foot, so to speak. So again, it's using the right tool at the right time, much like your rehab processes. You don't want to just jump straight into the most complex movements or the most strenuous movements, but they do have a time and a place.
Yeah, great coaching makes a huge difference. And I think the coaches who understand programming so well, it's incredibly deep, it's incredibly multi-layered.
Yeah, it's multifaceted. There's not only, you know, the actual movements, the biomechanics of it.
Yeah.
The technique is, you know, the coaches are the best reason to get one is because they're there giving you live feedback for the actual movements. But yeah, if they can understand some of the, you know, just some of the molecular biology underlying recovery of the muscle, what are some of the stimuli that cause the muscle to grow, then if they can pair those up with the appropriate training, get it into the right microcycle into your programs, that's when you can really start to accelerate the performance.
There's a holy grail that we're actually really aiming for.
That's exactly right. We're trying to work alongside as biochemists and exercise scientists. You know, we're trying to just provide a framework that can then be translated to a certain athletic population. But the important thing to understand is that science is a slow process. Science is very incremental. Oftentimes we're just trying to catch up with what people are already doing. And so if, you know, I see the debates all the time on Twitter and Instagram, evidence-based approaches, science-based, whatever.
Yeah.
I totally agree. I'm a scientist. You need objective, non-biased literature to defend what you're doing. But unfortunately, we don't have all of the answers.
No.
But the great thing is, like you said, coaches, they might not have scientific publications to their name, but they've probably worked with a sufficient amount of athletes, and they have, you know, training interventions that work, and there is some merit to that. You know, you can't deny when you have an athlete that gets gold medals from your programs. You know, you're keeping them healthy, things like that. So I think there is a fine balance between the art of training and the science. So yeah, I wouldn't discredit either or.
No, there certainly is an art to it beyond the science, and
yeah, absolutely.
Science underpins the art. That's really where we come to. So I always have this issue with the evidence-based work, is that there's too many variables that often can't be accounted for and are missed.
Yeah.
And I often would say that's why the science to me is so important, because we have the underpinnings of the gold standards, so we at least know that if the research conflicts with the gold standards, then it's unlikely that the gold standards are wrong.
Yeah, no, that's right. That's a really good point to make as well. The other point to raise is the evidence-based approaches are great, but they're limited to the populations that we can study. So it's very rare that you are able to study elite athletes, especially even more rare in the strength sports, for various reasons, for funding and things like that. I mean, it's quite limited. So when you have these high caliber lifters talking about evidence-based approaches, a lot of the evidence probably that they're working off of is based off your average punter, which again, it still provides some evidence and some framework to build upon, but understanding how to apply it to your particular athlete is the really important thing. And that's what sets apart good coaches from great coaches. The great coaches can understand the research and the literature behind it, and they can tailor it to fit their athletes appropriately.
It's huge. And this is where you really have been, you're going into it, you're going to the satellite cells. As a stem cell therapy in my area of rehab is becoming a hot topic now. And it looks like it's going to be an area which there's going to be a lot more money invested into.
Yeah, I think stem cells kind of have that like ring to their name. Everyone hears about them, and they just kind of think, well, this is the magic cell. It can do everything. It can do anything. And yeah, in our laboratories, in vitro, when we grow them in petri dishes, they can do anything. That's right. They can do whatever you want. But as far as the in vivo, inside your body physiology, it's still kind of a black box, and they're quite a mysterious cell type, and that's more and more what the research is showing, and we're just trying to understand how do they behave.
So you can really observe them well in a petri dish, but once you put them in the body, it's going to be a different process.
Yeah, and it goes back to what we're saying. In a petri dish, we can control the environment perfectly. We can give the whatever desired amount of amino acids, sugars, carbohydrates, and we can give different growth factors. We can stimulate them to specific degrees. But in the body, I mean, there are hundreds and thousands of different variables. There's so many different metabolites. There's so many different proteins that all can converge upon the function of these satellite cells, and in particular, satellite cells in the muscle. And the process of them actually making their decisions to contribute to muscle growth or to actually go back into a resting state, we're still not, the research is not conclusive about how that happens.
So it's a great field for you to be in. It's obviously going to be super exciting, and there are people who are going to be obviously seeing these interviews who are looking at their careers at the moment. Perhaps it's high school and perhaps it's undergraduate, and they can really see that this is an area to which there is a lot more to come.
Yeah, absolutely. I mean, biomedical research or research in general, just stay curious. Just be interested in how stuff works. Ask questions. Don't be afraid. I mean, I was always, during my undergraduate, I'm not going to lie, I was always, at the back of the lecture hall, kind of was afraid to ask any questions, but you just got to put yourself out there. There's no bad questions. You're only going to learn more by asking the questions. And I think
I always say that, look, there's no such thing as a stupid question.
Yeah.
There's just stupid people.
That's right. There are absolutely categorically stupid people, myself being one of them for the most part. But no, stay curious. Research is a great field to be in. It's very self-rewarding. You go in, you can design experiments, you can learn how things are functioning, and you can apply them to different different fields. I'm an exercise scientist and I do a bit of biochemistry, but that goes beyond just training and powerlifting and sports and competition. I mean, you can get into drug discoveries, you can get into cancer therapies, you can get into biomedical engineering, prosthetics. All these things have a basis in understanding physiology, understanding how the body works, so then we can actually use technology to then assist in whatever the ailment might be or the remedy might be. So
it's an exciting world out there, Super Bobby. Well, thanks for coming in today.
Yeah, thanks for having me. It's been great.
And your journey is still going.
Yeah, so
on the way, lifting bigger, lifting better, and Alex Deacon there looking after your program as always.
Yeah, and just staying injury free, staying out of trouble. So
you got all the parts there.
Yeah,
Thanks
that's
for coming
right.
in.
Thank you.